Today is 23rd of September 2025. Uh, the biology, the test that deliver medicine to inside human body, certain body is really difficult because first there could be side effect and second, it could be just diminished while you're going inside there. But there's AI generated MLH, one small binder improves prime editing efficiency cell, which is about CRISPR is basically what does coding DNA and including double strand break. And prime editing is 2019 by David Liu. It became famous search and doing the replacement. Prime editing uses modified CS9 enzyme to block some reverse transcript enzyme because enzyme is going to block it, but prime editing is going to prevent it. That's what they're doing. And the problem is the cell has a natural proof reading system called the mismatch repair pathway to fix such mistake. The key protein that leads the charge for the MMR pathway is called MLH1. And also, September 21st, using CRISPR killing 14 desatragene when there's gene law. This is some of the very few law in biology about gene law. And Watson critics Craig in 1953, they opened the molecular biology era and central dogma, which is from DNA to RNA and from RNA to protein. This is really important. DNA to RNA transcription and RNA to protein by translation is also important. There's some Richard Lansky, which did the long-term evolution experiment, and there's approach of AlphaFold, and there's another approach which is directed evolution using selection, mutagenesis, transformation process, expression, and screening selection mechanism. There's selective breeding and DNA, which is cutting and put together after reading DNA sequences. So this is important. A human genome project, it was at 2003. John Craig Venter is famous, and micro's genome, which is making some biology, which to eat, then output of eating is just and data splitting. One way synthetic biologists make sense. Uh, Something in between these are interchangeable. Maybe it's because of information gap problem, or maybe it's because human DNA has, we are saying there's 3 billion pair. And for duplicated lamp, it was on 1996. It is Indiana level biology. We did some generation. And there's Genobot, which is on 2020 that using heart cell, and 2023 that do self reproducing. And 2020 finally, there's CRISPR clustered regularly interspaced short palindromic repeat genome editing technology came out, which is based on programmable RNA guided DNA nuclease. After DSP, there's some CRISPR development, which is applying for another biology and synthetic variation by Liu. After that, gene drive by using Mendel law. This is amazing because if you do it to one generation of mosquito by the gene's law, even in next generation and the other generation, there will be mosquito who has such gene that there will be distinct at one point. Yeah, and biology, what you can do is basically uh, there's DNA pathway, and by using prime editing, we are suppressing it and make new global molecule. This is very big thing in biology. Thank you.